in my lab studying one of the interesting complex called exon junction complex. So my lab actually interested in the neurodevelopment and how the neurodevelopment associated with all kinds of neurological disease. So we all know actually the brain actually started with one single layer of the neuroepithelial cells. We actually also call this layer of cells as a neural stem cell. So this layer of the neural stem cells will undergo divisions, asymmetric division to proliferate, and also asymmetric division to produce neurons. And those neurons actually will migrate towards the outer layer to form the giant network in our brain. So it's a beautiful network. And my lab actually interested in how abnormal neurodevelopment will lead to the neurological disorders. So one particular protein we are interested in is actually the exon junction complex. So we all know actually the transcriptomes actually start with DNA, then it will transcribe to the mRNA, but post-translational modification is actually very complicated. So actually we start with a pre-mRNA, then we'll undergo the splicing and the modification, export to the cytosol, then it will translate into protein. Eventually, the mRNA will be degraded by also very complicated process. So the exon junction complex include the RBNA, EIL4A3, BTZ, and MAGO. This complex actually involve many steps of this regulation. First, actually, it's bind to the exon junction and work together with the spliceosome to help the correct splicing. And then it's actually associated with the exporting and the importing will help mRNA to export into the cytosol. It will also interact with the translational machinery to stimulate translation. And eventually, this is actually have been well established. It's involved in the one of the evolutionary conserved degradation pathway called the nonsense-mediated mRNA decay. So how this actual protein involved in the mRNA degradation? So this pathway actually is actually interesting. It can detect any most of the nonsense mutation. So we know actually if the protein has the nonsense mutation, will produce the truncation or deletion. It will eventually impair the cellular function. So how does the exon junction complex help to detect this nonsense mutation? So we know actually if the mRNA will transcribe to prime mRNA and splice into the mature mRNA, the exon junction will sit on the every single exon junction. And the first round, the ribosome will kick out most of the exon junction before it's reached the normal stop codon. So if there's any premature stop codon, what you can expect actually, the ribosome will hit first premature stop codon before the last stop codon. So at that time, they still have the exon junction complex sitting in this mRNA. So the exon junction complex will interact with the ribosome at the first round translation, will initiate the degradation pathway. So this is how actually the exon junction complex can involve in the NMD pathway to protect cells from the nonsense mutations. So this is actually a very conserved pathway. And interesting, those proteins actually involve some of the disease. One of the disease we know is actually a mutation have been found in the recently published in the Nature Genetics. Is some mutation in the RBNA genes cause this thrombocytopenia and the absent radius syndrome. So this is actually a rare syndrome, but the patient with this mutation normally develops the short arm of the furling development is abnormal, and also they have the low platelet number, so it's called the thrombocytopenia. So you can see the mutation has the normally have the one of the 1q21 deletion, big deletion, and some mutation in the other chromosome that actually lead to reduce of the RBNA protein. So this basically is a haploinsufficiency or the recessive mutation. So this patients also actually associate with other abnormality. For example, they have defects in the kidney and also cardio abnormality, and also mental retardation and abnormal brain development. So the first thing we look at is actually how this gene involved in neurodevelopment. So if you don't have any background on the neurodevelopment, here is actually how the early embryonic development happens. So in the early embryonic stage, the new stem cell will divide and also proliferate. Then those cells actually sometimes undergo the asymmetric division to produce new progenitor cells. So the neuron, immature neuron, will migrate along this track outwards and form this actually inside-out layer. So first, the cells, first layer produce cells in the inner layer, and then it's produced the outer layer. So this is the inside-out manner. Then the stem cell actually still reserved in the ventricular zone and subventricular zone, keep dividing until the adulthood. So basically, we still have a residual neural stem cell in the adult brain. So first thing we look at is actually how the RBNA expression during neurodevelopment. So we isolate the brain lysate from the early E9, the embryonic day 9, all the way to the adulthood. Actually, we see interesting patterns that actually RBNA is highly expressed in the early embryonic stage and then decrease. So conversely, you can see the SOX2. This is actually the early embryonic stem cell marker. It's decreased correspondingly. So the neuronal marker TUJ1 increased. That means the cells actually start to differentiate into neurons. So also we look at the staining pattern in the early embryonic brain at E16. We know that actually stem cell actually localized in the ventricular zone and subventricular zone, and then the neurons actually localized in the cortical place on the top part. So the neurons with co-stain with the RBNA, this our interest mutation protein, and then the neural stem cell marker nesting. So you can see the RBNA is highly enriched in the neural stem cell population, but actually decrease when they differentiate into neurons. So one question, so how does RBNA, why does RBNA actually express new progenitor cells? What's the function of the RBNA in the neural progenitor cells? So we have one very unique technology. We can actually knock down and overexpress the plasmid into the embryonic stage called in utero electroporation. So basically, we can inject plasmid into the embryo brain in the ventricle area, then apply the small current to deliver DNA into the embryonic brain. We can put the embryo back to the mother, pregnant mother, and let them survive for whatever you want. Normally, we start with E14 and then sacrifice at E16, so basically 3 days. We can monitor the cell proliferation, differentiation, and migration. So here is basically one of the example. We can see we designed 2 shRNA knockdown RBNA, and in the cells, we can detect the knockdown effects. And then we look at how the RBNA knockdown affect the migration. You can see the cells in the control shRNA electroporated brain is distributed evenly in the 3 layers of the cortical plate, intermediate zone, and the subventricular zone. However, when the RBNA knockdown, we can see depletion of the new progenitor cell in the subventricular zone. So it showed the quantification here. And also, it's increased the population in the cortical plate. So this data suggests actually knockdown of RBNA promotes the neural migration and deplete stem cell pool in the early embryonic stage. So we further look at how this actually happened. Actually, we found actually we used BRDU as a marker. So the BRDU actually will incorporate into DNA only at the S phase of the cell cycle. So this is actually, we can label the progenitor cells. When we look at the, so the green cells actually will indicate that the cells are knocked down RBNA. So we quantify the BRDU percentage in those progenitor cells. We can see the BRDU incorporation is significantly reduced when the RBNA is knocked down. Furthermore, we look at the differentiation, look at the neuronal marker, the TOJ1. We found that TOJ1 actually increased, positive cell increase. So we hypothesized that the RBNA is actually critical to keep the balance between the neural stem cell and the neuronal cells. So it may be critical for the cell cycle regulation. To test this cell cycle regulation, we actually use a cell cycle exit index. Basically, we inject BRDU to the embryo and label the cells for 24 hours. And then we use the Ki67. This is actually cell cycle marker. Any cells still present in, still remain in the cell cycle will label with the Ki67. So any cell actually exists cell cycle will be Ki67 negative cells. So we basically measure the GFP positive cell and BRDU positive cell in the whole population at the beginning. And any cell actually exists cell cycle will be Ki67 negative cells. So when we look at the cell cycle exit index, we can see the RBNA knockdown actually increase the cell cycle exit. So basically, the RBNA is required to maintain the stem cell in the cell cycle. So conversely, we also look at the overexpression pattern. We see that actually overexpression stimulates the proliferation, stimulate BRDU incorporation, and suppress the neuronal differentiation. Consistently, we also see once we overexpress RBNA, it's reduced the cell cycle exists. So basically, RBNAs require the progenitor cell proliferation. So this is consistent. Furthermore, we actually make the condition knockout model of this gene. So we flux the internal 3 exon of this RBNA gene and can see once we infect the fibroblast cells with the CRE, we can completely knock out the RBNA in the cells. And strikingly, when we cross this flux mice with the nesting CRE, that's actually the neural stem cell specific CRE line, it will wipe out, actually delete one copy of the RBNA gene in the neural progenitor cells. What we see is actually you can see this is a smaller size, and also the length of the size, the mice is smaller, and also the weight is significantly reduced. And also you can see here, this is actually P17 mice. Let's see. Okay, they can move around normally. But actually the knockout mice is completely paralyzed. So they cannot move. Eventually it will die at postnatal P20, around P20. So when we look at the brain size, consistent with our data, actually the brain size is significantly reduced. You can see this is a very dramatic effect. This is only one copy of gene, still have one copy in the brain. And the proliferation, we look at BRDU, you can see the BRDU labeled cells significantly reduced. And also, after we dissect out the P12 mice brain, you can see the dentate gyrus. This is the dentate gyrus in the wild type mice. this dendrites of the knockout mice is severely reduced. The cortex also show the cortex thickness is reduced. You can see I talk about the different cell layer. The layer is completely screw up. We use the specific marker for the COX2 is actually labeled the layer 2-3 marker. You can see the COX2 is very good labeled the layer 2-3, but actually it's just dispersed in the knockout mice. So this is the indicated the neural layer defects in the RBNA knockout brain. So furthermore, I want to use actually this is how the transcriptome analysis help us to determine the underlying mechanism for the RBNA mediated effects. So we look, we overexpress RBNA in the human neuronal cell line, and we look at how many RNA actually changed. So this is about 7% of the transcriptome actually affect, and mostly the protein coding RNA. And also I found some of the link RNA, the non-coding RNA, and sense RNA, and the stop RNA actually change, but this is only account about 5% of the total upregulate or downregulate genes. But when we actually cross-reference with the disease-related risk gene, we found actually among those transcripts, the risk gene for the autism spectrum disorder, schizophrenia, and also the Alzheimer's disease and the intellectual disability are highly represented in this pool of the genes, but not in the Crohn's disease. This is related to the other system. So, we also look at some of the gene actually in the protein level. Consistently, we found actually the neurolegum 1 is actually reduced in the overexpression, and the KIF1A, which involved the neural migration, increased with the overexpression. They downregulate when the RBNA is knocked down. So this is actually show a consistent result of RNA-seq data at the protein level. So we also look at the functional cluster. Actually, we see actually different function actually indicates in our transcriptome analysis, particular cell proliferation, differentiation, and also the pathway that's related to proliferation and growth factor signaling and role signaling pathway. Particularly, I'm surprised actually we can see some of the function is actually related to behavior. And so we go ahead actually test some of the gene function of the RBNA in the behavior. So this is actually give our future direction. So we look at the RBNA in the adult brain is highly expressed in the dendrites and the hippocampus. This is associated with many brain behavior. And we also see this, the RBNA expressed in the dendrites, although it's low, but actually we can still see it in the dendrite and the nucleus. Then we look at the behaviors using the stereotactic injection. Basically, we can overexpress the protein in particular region of the mouse brain. So we use the GAP-labeled RBNA, inject to the dendrites. We can see very good expression. We look at the behavior in the open field test. So basically, we put the mice in the open box and let them run. And we found that there's actually significantly reduced time in the center part. It's indicated they are less anxious because normally the mice or the animal, they try to hide in the bushes, and also they try to avoid the center part because that's where the predator can catch them. However, we can see the overexpressed RBNA, actually they spend more time in the center. And also we look at the elevated plasma, we see that actually they have reduced the number into the open arm, so it indicates some anxiety behavior. So to summarize this talk, actually we found RBNA actually stimulates proliferation, and suppress the maturation, and also involve some behavior. So in the future, we are actually we'll look at more detail in the other organ and how it's actually regulate this process. So finally, I want to thank my lab, the graduate student and a scholar to finish this work. So the Emma is actually the visiting scholar responds to behavior test, and Colleen also involved in the all the transcriptome analysis. So also we have a wonderful collaboration in the Penn State. So Dr. Albert helped us some of the bioinformatics too, and Dr. Lucia helped us with the behavior test. So we have also other collaborators in UC Irvine in the Edge. Thank you very much. So any questions?
Okay, we got about a couple, maybe you could take a couple.
Thank you. Dr. Mao, it's a nice presentation.
Yes,
so I just wondering, because this RBM8A is an essential factor for the exon, junction complex.
Yeah.
So what makes this more, responsible in the brain, instead of in other tissue?
Yeah, that's a good question. Actually, I think this is actually mystery. Actually, in some of although those exon junction complex work together, but the human disease, at least human genetics, they found that the mutation in different complex factors actually cause different disease, different syndromes. So we actually don't know. Actually, one aspect actually they have particular or specific function in different tissues. That's I can imagine. So maybe they interact with a different factor in the tissue and control different substrate of the transcripts in different tissue, because although they are essential, but the expression, you can see the expression level is different even within the brain, right? So they basically they probably have different function in the neurons, different function in the neuroprogenitor cells, different function in the macrophage. It's highly expressed in the blood system, so that's why the deletion actually caused the low platelet number. So
you think that tissue specific difference probably come from cofactors?
Cofactor or specific substrate. So they
expression difference.
Yeah, they actually, when we also did some of the RIP assay, it didn't bind every single transcript. So although it's they call exon junction complex, but actually it's not bind every single transcript. And some of the transcript only have one exon, so it will not bind those. So it's not substrate. Okay?
Well, I think in the interest of time, we need to move on. So any other questions, please take offline. Let's thank our speaker again, please.
Thank you.